Validation Test Analysis Presentation. Hello, good afternoon. My name is Alan Esquivel and I will be presenting this presentation on validation test analysis of an instrument or method in forensic science, reviewing both instrument and method in which both are used in forensic cases like missing persons, sexual assaults, and homicides. Though recognized as a recent advancement in forensic technology, it has shown some promises in expanding region of DNA analyzation. Instrument validation and methods. The purpose of validating instruments and methods before using them in forensic practices is to ensure the validation confirms that the instrument or methods are consistent and produces dependable lab results. It demonstrates if the performance is adequately suitable for the intended use in forensic application. Validation also helps in discovering any weaknesses, constraints, or circumstances in which the instrument or method may fail. Proper validation enforces the support and the admissibility in legal court by providing technique or method is scientifically backed, giving the impression that the different forensic analysts or laboratories can reach the same examination conclusion using this validated process. Overall, validated tools can contribute to quality assurance by reducing errors and enhancing confidence in forensic examinations for legal court use. Developmental validation of the Powerplex Fusion 6C system kit. The Powerplex Fusion 6C system kit is designed to amplify 27 Loci in a single PCR reaction with the use of 6 fluorescent dyes. This type of system improves the ability to discriminate mixture interpretation and database compatibility in analyzing complex or degraded DNA evidence samples. The Powerplex Fusion 6C system kit is engineered to be compatible with the modern capillary electrophoresis instruments like the ABI 3500 genetic analyzer and is used for forensic DNA extraction from a wide range of standards and quality of biological evidence. Widely used in forensic casework, databasing, and person identification because of the robust sensitivity and reproducible results that meet international standards for forensic DNA analysis. Identifying all 27 loci. A rough explanation of what these 27 loci represent and identify in DNA evidence is starting with the 20 autosomal loci in part of the CODIS core loci. CODIS standing for Combined DNA Index System that contains the DNA profiles contributed by federal, state, and local participating forensic laboratories. NDIS is a national DNA index system is considered one part of the CODIS at the national level. Auto STRs are inherited from both gene parents that are used for identity testing and matching evidence in forensic databases. In addition, with 3 autosomal STRs, Penta D, Penta E, and SE33 enhances the capability to international databases and discrimination competence. The 2 short tandem repeats, or STRs, DYS576 and DYS570, are found on only Y chromosome and are used to distinguish male DNA in a mixture or degraded evidence. Lastly, amiglogen and DYS391 identifies the X and Y chromosome that assists in sex determination on a DNA sample. Validation instrument or method. The Powerplex Fusion 6C system kit functions by using a polymerized chain reaction or PCR to enhance specific regions in the DNA, simultaneously amplifying the 27 different genetic loci in a single reaction tube that greatly improves efficiency, accuracy, and differential technique in DNA profiling, following the steps of 1. DNA extraction, processing starting with the extraction of DNA from collected forensic evidence like blood, saliva, or biological stain. The sample is purified and quantified to guarantee the quality and acceptable amount for the next step, PCR amplification. Step 2. PCR amplification. With the use of PCR primers or short strands of DNA to target specific 27 genetic markers, the primers are organized with the fluorescent dyes for recognizing the results in later steps. Step 3. Fluorescent labeling. Tagging the paired primer with different colored fluorescent dye that reveals the fragments for detection of locus in the chromosome. Step 4. Capillary electrophorese occurs after the PCR amplification that separates the DNA fragments by size with the use of capillary electrophorese, or CE. Injects the fluorescent labeled DNA fragments into a thin capillary tube filled with gel-like polymer. Then an electric current is put in that will migrate smaller fragments faster than larger fragments. Step 5. DNA analysis. Results from the electrogram is analyzed by the use of specialized computer software to distinguish the alleles present at each locus. After compiling the DNA profile, it is compared to the CODIS, aka the Combined DNA Index System, that contains the DNA profiles contributed by federal, state, and local participating forensic laboratories, concluding with the displaying the ability to provide detailed and accurate DNA profiles from a limited or degraded biological evidence, making it a powerful tool in modern forensic science. Applied Biosystems 3500 Genetic Analyzer. The ABI 3500 Genetic Analyzer is a capillary sequence that is able to read DNA samples and generate an electronic file with symbols that represent sequences of nitrogen bases like adenine, cytosine, guanine, thymine, and uracil of the human genome. Also recognized as a multi-capillary electrophorese instrument, the ABI 3500 genetic analyzer is devised to separate genetic strands based on size and inputs the data into a specialized computer software for interpretation. Most examinations are from biological samples that are denatured and leaves DNA single-stranded. The ABI 3500 genetic analyzer is capable of sequencing nucleus, mitochondrial, and as well as plasmid in the whole blood. The application is that it could run a wide variety of de novo sequencing, resequencing like mutational profiling, and microsatellite analysis. Tests performed. Validation of Powerplex Fusion 6C system is a series of design tests that align with the standards of quality management and forensic science, specifically the Scientific Working Group on DNA Analysis Methods, or SWGDAM guidelines, and the ISO IEC 17025 accreditation standards. The tests are crucial to guarantee that the system meets the expected standards in forensic DNA analysis, starting with the sensitivity testing that ensures the examination of reliable results even from the smallest or degraded samples that are especially common in forensic casework, following with the precision and reproducibility testing, which is a common component of quality management as they demonstrate the process is controllable and sustainable, that minimize error factors of either human or technical aspect. Next is the mixed studies, reinforcing that the kit can distinguish and interpret overlapping DNA profiles, a crucial quality requirement for forensics validity. Going in hand with concordance testing is a database compatibility in the continuation of forensic casework, accomplishing the quality assurance needed for consistent and traceable results across a variety of methodologies over the decades of different instruments, or STR kits. Continuation of tests performed. In forensic evidence, contamination is expected, and it can be due to a number of factors. That is where inhibitor tolerance testing is key, by evaluating whether the Fusion 6C system kit can still function under such conditions. Focusing mainly on the accuracy is correct regardless of the state the quality sample is present. With the stutter and artifacts evaluation, reinforces neutrality and sensibility of DNA interpretations as necessary in meeting criteria for accreditation standards, while robustness testing falls under the unavoidable variations in laboratory practices that is a rudimentary aspect of sustaining consistency and quality in forensic casework practice. Lastly, the species specificity test and quality management to prevent any non-human examinations from affecting forensic analysis results. Validation test analysis. With the Powerplex Fusion 6C system kit being properly validated and meets forensic accreditation standards. Summary of validation. In the sensitivity testing, the Fusion 6C system kit performed well as expected even with minimal DNA input. For precision and reproducibility testing, it generated profiles matching previous Fusion 6C system kits and verified the allele sizes within specified tubes with little to none size variation across a number of individual instrument runs. The mixture studies evaluated the success in allele separation from a variety of mixture ratios. No indication of allele call discrepancies in the concordance testing, In the inhibitor tolerance testing, the kid reacted in the presence of inhibitors such as hematin and humic acid. Stutter, artifacts testing, and robustness remain steady when reviewing signal variation and artifacts like stutter peaks, and demonstration and verifying for the high specificity for only human DNA in the species specificity test. Analytical gaps and added improvements. However, there is analytical gaps present in the Fusion System 6C kit, like unbalanced mixtures, that has unknown limitation in mixture interpretation. This may involve multi-contributor mixtures which go beyond two individuals' DNA present in a single sample, as well as the lack of knowledge on identifying a low-level contamination of DNA evidence, if the kit is able to differentiate minor contamination. Though an uncommon occurrence of rare allelocystatic event, testing of the kit is needed for such situation. Lastly, the performance evaluation of the YSTR and mixtures. A broader understanding is needed on the YSTR capabilities of detection. Added improvements. In the Powerplex Fusion 6C system kit should include a more real-life simulation and environmental effects or challenges to validate further, including a comparative performance study to provide feedback in comparing the performance of other competing STR kits, examples being NGM Detect and Global Filer. Finally, as labs are increasingly developing probabilistic genotyping computer software to keep up with immense caseloads, the importance of seeking a program of automation interpretation can be integrated on the Powerplex Fusion 6C kit analyzation to further improve output efficiency. Conclusion. In the final thoughts of Powerplex Fusion 6C system kit proved while undergoing the rigorous process of validation testing, it revealed a superior performance across the board, proving extensive forensic capabilities in sensitivity and accuracy on extracted DNA from degraded evidence samples. However, areas in performance like the environmental stress testing and the integration of automated interpretation computer software would represent an excellent opportunity for a higher validation in the forensic community. Viewing real-life obstacles as mentioned before and having improved efficiency if automated interpretation is implemented will enhance the forensic professional field and having confidence in the Fusion 6C kit's utility and the way forensic laboratories conduct and approach diverse challenging forensic casework. References. Please feel free to pause the video and review my references for this presentation. Thank you for your time and attention.